So before I start my talk, I'd like to acknowledge the traditional owners and their custodianships of the lands on which we meet today and pay my respects to the elders past, present, and emerging. So my talk today is about my PhD project on cryopreservation for conserving Gossia species threatened by Myrtle in Australia. And I'd like to start by giving a brief introduction to the genus Gossia. So Gossia is the main genus in Myrtaceae, has been reported for having this very special trait, which is metal accumulation, with Gossia fragmentissima being a multi-metal hyperaccumulator, and it can accumulate manganese, copper, and zinc. And many Gossia species are now severely threatened by metal rust and face a high risk of extinction. So in Australia, according to the EPCS, both Gossia fragmentissima and Gossia glaucata are listed as endangered, and Gossia hilii is now also under assessment. And more Gossia species are recognized as under threat at state level. So in response to the infestation of myrtle rust and also the emerging environmental threats, it is necessary to develop ex-situ conservation methods. Usually seed banking is a primary ex-situ method, but it does not readily can be applied on all Gossia species. This is because that some of the Gossia species produce few seeds in the wild. One of the reasons for that is myrtle rust. So because myrtle rust can attack on the flowers and fruits, therefore causing the problem of seed production. And also, many of the Gossia species have fleshy fruit, and the seeds are likely to be short-lived in seed bank conditions. Therefore, under these circumstances, alternative ex-situ methods such as tissue culture and cryopreservation are urgently needed. And my talk today is going to focus on the species Gossia fragmentissima, and hopefully this can be used as a case study to talk about the future ex-situ conservation options for the threatened Gossia species in Australia. So I'll start from tissue culture. So tissue culture basically refers to the sterile growth of plant cells, tissues, and organs. And this diagram here is some basic steps in the tissue culture protocol. So starting from the establishment in individual cultures, multiplication, routine, and implementation. So this is commonly used in plant propagation, but it can also be used in short to medium term conservation. So for Gossia fragmentissima, the Australian Plant Bank has firstly established some individual cultures and developed this MS-based multiplication media. MS is basically one of the most commonly used commercial plant culture media, and they have also currently shared some with us for further cryopreservation development. But while we were trying to maintain and multiply these cultures, we've noticed that the health conditions in the cultures declined. And we tried to switch the basal media from MS to the media, which is another commonly used plant tissue culture media. And the conditions improved a little bit, but some problems such as fertility still remain. So we were hoping to further optimize the basal nutrients in the media, and hopefully we can further optimize the culture condition before we do any cryopreservation development. So here we've used a method called response surface methodology. So traditionally for media optimization, usually a bunch of different media are developed relatively randomly, and these media will be prepared, and hopefully one optimized media can be selected from the tested media. So this method usually requires lots of material, it is time consuming, and not always successful. But for response surface methodology, these media formulations are created based on a certain design, and instead of just compare and select one from them, we've used those results for advanced statistical modeling, and then one optimized media can be predicted based on the constructed models. So this method usually requires less material and less time, and the results are usually more reliable. So in this experiment here, we've selected 6 nutrient components in a woody plant media as the factors that we want to adjust using responsiveness methodology. And these numbers here are the concentration ranges we tested. And by using the software design expert, we've generated 14 media formulations using these factors. And we've also set up some criteria to measure the cultural responses towards these media. So the whole process took us about 3 months. So after that, we collect all those results and constructed models. And then again, using the software, we generated this predicted best media or PB media. So the nutrient compositions in the predicted best media are shown here using red bars, while the nutrient compositions in woody plant media are shown by the purple bars. So as you can see, there's a huge increase in ammonia nitrate, mesols, macros, and EDTA, and there's a decrease in calcium nitrate. And after we generated this media, we've also compared it to woody plant media and also 2 of the best performing media among the 40 media we tested earlier, which is media 15 and 25. And we've confirmed the superiority of media, best media and supporting better responses for Gossia fermentissima cultures in sample counties compared to woody plant media. So moving on to cryopreservation. Cryopreservation refers to the long-term storage of the regenerable plant tissues in ultra-low temperature, usually in liquid nitrogen. The key thing in developing cryopreservation protocol is to ensure there's no formation of any ice crystals, which would rupture cell structures and decrease cryopreservation success. This is usually achieved through humidification of cellular moist. And there are many different cryopreservation methods used for cryopreservation, but the one I used here is called droplet vitrification. And this diagram here is some basic steps in a typical droplet vitrification protocol. And this is usually start from the source of exons from donor plant and explant excision, and then these explants would be treated with loading solutions for osmotic protection before they get dehydrated in vitrification solutions. After that, these explants will be placed on an alcohol strip in drops of fresh vitrification solutions before they got plunged in liquid nitrogen for storage. And for regeneration, these explants would be rewarmed in unloading solution and then regenerated on replate and then reverse media. And sometimes these donor plants or the explants will also be pre-treated. This is usually used to help them better cope with the following cryopreservation procedures. And the pre-treatment I use in my protocol is sucrose pre-treatment. So place explants on sucrose containing media and only allows the explant to dehydrate a little bit before the actual cryopreservation procedures, but also allow the natural cryoprotectant sucrose to accumulate in the explants. And theoretically, this would help the explants better tackle with the following dehydration and cold stress in cryopreservation. And currently, cryopreservation protocol is still species specific, which means we need to optimize these single steps in order to make it work for different plant species. So as you can see, it's a multi-step protocol. So it's quite common to set up these controls such as dissection control, DC, or loading controls. This will help us to understand the impacts of individual steps on the viability of the explants. So I've done quite a lot of optimization of the protocol earlier on like the selection of explants or the loading treatment time or the selection of vitrification solutions and treatment durations. But today I'm only going to share some key experiments I did before actually this working protocol for Gossia fermentation. So as I mentioned earlier, I've incorporated sucrose pretreatment. So in this experiment here, I've tested several different concentrations of sucrose pretreatment. And I've also used shoe tips from both predicted best media and from impact media. So let's first just look at the results of the shoe tips from predicted best media. So here I've compared three concentrations of sucrose pretreatment, 0.2, 0.3, and 0.4 molar. And this 0.009 molar is a control, which is the same concentration as is used in the application media. And if you just look at the light gray bar, which is the regrowth of the shoe tips after liquid nitrogen storage, as you can see that they all achieved fairly high post-cryo regrowth with or without sucrose pretreatment. And this one is results for shoe tips from wheat plant media. And again, if you just look at the light gray bar, which again, there's no significant difference among the different pretreatment groups. So from these results, it suggested that sucrose pretreatment, at least for the method and the concentration range we tested, did not improve post-cryo regeneration for Gossia fermentissima. And in this diagram here, I've combined those two results together. So this is a direct comparison of the shoe tip regrowth of after liquid nitrogen that was originally from predicted best and wheat plant media. So you can see that shoe tips from predicted best media supported significantly higher post-cryo regrowth with or without sucrose pretreatment. So these results suggested that predicted best media improved post-cryo regeneration of Gossia fermentissima, and this is likely due to that predicted best media supported better health conditions of the cultures before doing cryopreservation. So once we've achieved this working cryoprotocol for Gossia fermentissima, I've also tested it across different accessions of cultures. So all the previous experiments I talked about were using the accession 01, and here I've used accession 0124, and both of the two accessions were maintained on the predicted best media. So as you can see from the diagram, we can achieve quite consistent results with both accessions. Although that for accession 0124, it seemed that sucrose pretreatment of 0.4 molar supported significantly higher post-cryo regrowth compared to control. So now we've achieved the working tissue culture and cryopreservation protocol. We also wanted to see whether this lab-grown plants can survive or grow under more natural conditions. Therefore, we carried out a small-scale planting with both in vitro and cryo-derived plants. So as you can see from the photos here, after bleaching and the concentration, we've now planted some of the plants in the garden near our lab. So these are the three types of Gossia fermentissima plants established in the garden. The first one is just an in vitro derived plant. The second one is a vitrification solution treated control, and the last one is a cryo-derived plants. So at least visually, we didn't see any morphological difference across the three types of plants. So this again suggests the idea that cryopreservation did not negatively impact plant development. So some key takeaways from my talk. The basal nutrients in the media significantly impact cultural growth. And we've developed an optimized predicted media using responsiveness methodology, and this may inform the future directions for optimizing other fertility media. And with collaborating with the Australian Plant Bank, we've developed a full tissue culture protocol for the species. We've also successfully developed world's first cryopreservation protocol for Gossia fermentissima, achieving 100% post-cryo regeneration, which is surprisingly high for woody species. And culture health is critical for cryopreservation, and optimization in tissue culture may reduce the required efforts to develop a working cryo protocol. And my project is part of this ARC linkage program, Cryobiotechnology Innovations to Help Fight the Motorized Pandemic, led by Ricardo from Curtin University. And I'd also like to acknowledge all of the collaborators and also thanks for New South Wales Department of Climate Change, Energy and the Environment and Water, and Australasian Plant Pathology Society for providing the treble bursary. And Leanne from the City Council, and our Haywood Coffee tissue culture team and my supervisors. Thank you.